tenemos que continuar con el programa. Y a continuación es la conferencia magistral del Congreso que va a impartir el profesor Pinzani, el doctor Pinzani. Yo tengo aquí la relación de todos los cargos que ocupa, lo que ha hecho el doctor Pinzani, pero utilizando el propio símil que acaba de hacer el doctor Ginés, lo primero que diré de él se dedica, su actividad es la investigación translacional en hepatología, es uno de los pioneros en el estudio de la fibrogénesis, en el estudio de las bases moleculares de la fibrogénesis, en los mecanismos moleculares en los que se funda la progresión de la fibrosis en las enfermedades hepáticas y más recientemente estudiando los mecanismos en que se funda la regresión de dicha fibrosis. También ha sido pionero y es pionero en el estudio de armas, de instrumentos, de herramientas no invasivas para el estudio de la fibrosis. El profesor Pinzani es catedrático primero de medicina interna, después de gastroenterología en la Universidad de Florencia, y hace ya unos años, en concreto en 2012, emprendió otra aventura como es dirigir el Instituto de Enfermedades Hepáticas y Digestivas y ocupar la cátedra Charles Sherlock de Hepatología en el University College de Londres. Máximo, muchas gracias por atender nuestro requerimiento para que vengas a impartir esta charla y cuando quieras.
Thank you very much for the introduction, Agustin, and I'm very honored to be here, and I was very touched by the presentation for Professor Rodesh, and probably one of the reasons why I am in London now as a Sheila Shello Chair is because Spain and Italy over the last 30 years have more or less taken over hepatology, and Professor Rodesh was a major promoter of this, together with other people. But I had the honor to work with him as associate editor for Journal of Hepatology, and he was a big mentor, at least for some time. So a lot of respect. And thank you for the invitation. I am very honored. And the title of this presentation is challenging, and basically I'll try to give you what is my vision and perspective, the way we should approach the search for targets for antifibrotic treatment. So we are all optimistic these days about the possibility of treating hepatitis C and hepatitis B, and we have big hope that if we can stop viral replication, we're going to have some major effect on fibrogenesis, and this is true, although we need to be careful. But once this is done, there are other chronic liver diseases which are characterized by fibrosis, and particularly NASH. NASH now is everywhere. I think I get like 10 emails per week because all companies are interested in fibrosis in NASH. There are other entities like PBC and PSC, which we don't know yet, what is the cause. So, any antifibrotic treatment will be very important. But also, now the tendency, the trend is to treat cirrhotic patients, because we know that if we can stop cirrhosis or even have some reversal of fibrosis in cirrhosis, this will have a major impact on survival, the need of transplantation, and the management of these patients. So actually a very important point is to treat with antifibrotic patients that are treated with antivirals in order to stop the kinetic progression of the disease that occurs even if we stop the virus for a few years. This is just like to show you the competitive landscape for treatment of NASH. Only partially, this is dedicated to an antifibrotic treatment, because the expectation is that if we can remove fat, we can remove fibrosis, which is not true. And also another expectation is that if you reduce inflammation, you can reduce fibrosis. It is also, not completely true. But just to tell you, there is a lot of movement. Many companies are active, and there is a huge investment, considering that, for example, the average cost of development for a drug that may fail is over $11 billion. So no jokes. And then, having said, these big numbers, and we look at if we have or not an antifibrotic drug. This is the search for antifibrotic drugs from phase 1 to clinic. There are different organs: kidney, lung, liver, heart, skin, and others. And you see the progression of potential drugs as antifibrotic. The liver, which is the purple, is very poor. There are very few dots, and actually the one that are right now, or they have been recently in a clinical trials like anti-LOXL2, losartan, pirfenidone, and N-acetylcysteine. It is amazing that none of these drugs, excluding LOXL2, which has failed, come from the 30, 40 years of research on molecular targets. These are just repositioning of old drugs. For example, pirfenidone, which is used in idiopathic pulmonary fibrosis, was introduced as an antiparasitic drug in 1956, and now is used as an antifibrotic. So basically, all our research is not at all translated into clinical trials. And these are, just to give you some example, some targets under evaluation. This is category is mostly related to collagen and myofibroblasts making fibrosis, integrin, TGF-beta. There is another chapter of targets which is close to PBC and PSC, bile acid receptor like FXR, like obeticholic acid, bile acid transporter, PPARs, nuclear receptor, cannabinoids, oxidative pathways, and another chapter with more dedicated to inflammation, toll-like receptors, cytokine, chemokine. So basically, there is a huge movement, all this based on 40 years of research, which is still stacked at the level of validation. So there is nothing that even goes close to a good preclinical study. And so basically, the problem is that no licensed antifibrotic drug after more than 40 years of active research. And what is the problem here? The problem is that we look for targets putting some cells, sometimes the cells are from a rat or from a mouse, on plastic. And then we do in vitro experiment, and then we think we have found a target, okay? Then, the target goes into an exponential number of mice or animals, and then this is actually a carnage of animals. And we know that no model is able to reproduce human disease. And basically, there is no translation. So there is all this. This is why it costs like 11 billion, because you could just insist on this, and we don't get to this point. In addition, we know right now that if you introduce a target for treating fibrosis, you also need to introduce an adequate biomarker for monitoring the effectiveness of the drug. So, this is actually becoming, almost a mandatory requirement. And so basically the question, just to stick into your mind, why we are not translating. So we basically have at least 99% failure rate when you try to go from animal models to humans. Animal models, I don't want to expand too much, but these are mostly mice, and there is no actually a single animal model, with the exclusion of some very recent model for NASH that has much affinity to human, and 9 to 10 drugs fail in human trials. So at some point, people like me that have dedicated many years to study fibrosis, and I don't hide that I have been using cell on a plastic dish, thinking that 20 years ago, we found all the possible pathway, and now we stop inhibiting, and we have basically failed. So just considering this is a hepatic stellate cell as a cellular target, what we should consider. So basically, my point is that instead of just considering the cell biology of the cell itself, consider the fibrotic environment. So whatever is around the cell and how much this can influence fibrogenesis. For example, hypoxia and neoangiogenesis are very important for the irreversibility of cirrhosis. At some point, the inflamed and fibrotic liver become very anaerobic, so is a different type of metabolism. We should use three-dimensional in vitro model. We should consider more the nature of the extracellular matrix that is modified during the fibrosis. And also, as a parallel to the use of FibroScan, for example, in clinical practice, we should also consider that tissue stiffness and contraction of fibrotic tissue are very important mechanisms. So what I've been trying to improve over the past 4 years, is the introduction of a different model, and just to explain to you what it is. So basically, you can make a three-dimensional model by culturing cell in some artificial scaffold. This can be made of collagen, polylactic acid, and you see they have basically these little holes, this space where you can culture the cells, and the cells, they are not anymore on a flat surface, but they are in a three-dimensional structure. Just to give you an example, basically, if you culture cell on a plastic flask, the cell are subjected to a stiffness of 2-4 gigapascal. And if you consider, a liver without cells, decellularized liver scaffold, the stiffness is 1-3 kilopascal, which is very much similar to what we detect with FibroScan. So this is the stiffness of the liver is due to the scaffold. And these are other, possibility which basically are closer to the reality than what we have been using, so far. So the point is that using plastic is very artificial. So this, the step, to go into a new modeling is called tissue engineering. This can be used for regenerative medicine, so to make a new organ or extracorporeal device. It can be used for disease modeling, so basically you recreate into a small piece of tissue the disease. So these are the cells, they go into the scaffold, which is repopulated, and you have, which is called engineered tissue. So how do we do this well? You need to have some good material. So I'm sure you know what happens to donor livers. In general, fortunately, 75 to 80% is transplanted, but 20-25% is rejected. And this is a good liver. I mean, is a donor liver. And at least what happened in the UK, this is because there is an excess of cold ischemia time, so it cannot be transplanted, or even more common, the liver is too fat, or there are some major morbidities found in the donor. For example, during the explant of the donor liver, they found that the patient has a colon cancer or another type of cancer, so this cannot be used. So basically, 20-25% of donor organs are rejected, and they go straight into the bin. They go into the garbage. So we have been setting a method, and I must say that the Royal Free had already a huge ethical permission from NHS blood and transplant to use discarded organ. And in November 2013, we achieved to make the first totally decellularized human liver, and this is Giuseppe, is my PhD. He was my PhD student, doing this decellularization using basically everything we found in storage, just old material, and this costed in the end a total of 1,200 pounds, And at the same time, in Harvard, they did a kidney, human kidney, with a grant of $20 million. So I think we were working in economy. This is Professor Max Malago, is one of our transplant surgeons who came during the night to fix some of the leaks, that were coming, so this was our first breakthrough. And since then, we do this on a regular basis. We don't use any more old material, but we have now a very nice type of bioreactor. We have two of these bioreactors here where we put the organ, and the organ is perfused. All the cells are removed. So in the end, we have the whole scaffold. We also have set a method for using portion of normal liver, like, for example, liver resection. We have put together a system using G-force, and we can make scaffolds directly from cubes obtained from the resection. And then we do bioengineering, we repopulate this scaffold, for disease modeling, as I will show you later. So our platform that we are proposing now is the following. We have donor healthy human liver, and here is the characterization of how the cubic scaffold looks like. These are all the different staining for collagen, and other extracellular matrix protein. So everything looks very nice, where it should be without any cells. This is a scanning electron microscopy. This is a portal tract. This is a portal vein branch, hepatic artery, bile duct. This is the liver lobule, and this is higher magnification. And this just to highlight, this is a space for one hepatocyte, into the scaffold. And it's amazing. It's the first demonstration that the interhepatocellular space is actually not like a basal membrane, but is like a basket of extracellular matrix protein. We also use explanted cirrhotic liver. We make a fibrotic scaffold. This is the fibrotic scaffold, and you see here how it looks like. This is a typical scaffold with capillarization of sinusoid, and just compare the normal hepatocellular space with the hepatocellular space in a cirrhotic liver to show that one hepatocyte, which is in this area, is unable to exchange oxygen or molecules, not as easily as you have in the normal situation. This is to show how we repopulate the scaffold. So each one of these wells has a cubic scaffold, and the cells are circulated into this bioreactor. And I hope you don't get a headache. And so basically there is a flow of cell that go through all these wells, and so the cell, they meet the scaffold, and they stick to the scaffold, and they start grow into the scaffold. The system is completely automatized, can be controlled also from home, with a smartphone. And let's finish this. Okay. And the result is this. This is a scaffold bioengineered with hepatocytes. So the hepatocytes, when they meet the scaffold, they start sticking on the surface, and then they penetrate inside. They make like situation similar like an hepatic lobule. This is like a central lobular vein. This is how they look like with scanning electron microscopy. Also, this is a comparison between albumin made on plastic in black and albumin made on a 3D system. You see, after 14 days, the cell on plastic, they die, so they don't make any more albumin, but they still make, actually they start to make a lot of albumin when they are hosted into the scaffold. This is another cell type, stellate cells, which are important for studying fibrosis. And you see how the cell get into the scaffold. They repopulate the scaffold, and they make collagen less than they make on plastic, and this is very important. And also they make LOXL2 when they are on the 3D, but they don't make LOXL2, the cross-linking protein, when they are on plastic. So the biology of the cell from plastic to the 3D system is completely different. So the question is that basically the failure of translating is because probably we have used models that are not really realistic for human biology. These are endothelial cells, so these are genetic endothelial cells. They penetrate into the scaffold and they go into the vessels by themselves, and they repopulate the endothelium of the vessels. This just to give you an example that I showed you before. Collagen on plastic is very high, and it's not as high, when you put the cell in 3D. The opposite for LOXL2, and looking at inflammatory cytokine, the cytokine are much more, higher production in the scaffold than on plastic. So, just other example of not matching the reality. This is FG2 cells which resemble hepatocytes, and this shows for example, the expression of alpha-fetoprotein much higher on the scaffold than on plastic. And this is the figure. So this is, if you take a hepatocellular carcinoma cell and you plate them on a healthy scaffold, this is the expression of alpha-fetoprotein. If you plate them into a cirrhotic scaffold, the alpha-fetoprotein expression is about 5 times higher. So the cell recognizes the environment where they are. Okay, so the solution is, as I said before, bioengineering. So we have started making models, putting together hepatocytes and extended cell, for example, and basically the proposal is, instead of going straight from plastic to animal to human, start looking at the target on a 3D system, then use animal model in a limited amount just to highlight the in vivo situation, and even to start looking at the efficacy of the drug, then go back for toxicity and efficacy to a 3D model, and then look for antifibrotic and biomarker to be translated into human. We have, for example, developed the first all-human NAFLD 3D model by using perfusion with oleic acid and palmitic acid, and you see that the cells slowly, they start accumulating fat, and it's amazing that the cytokine production is very similar to what you see in the human situation. Another important point is the biomarker development. So from this scaffold work, we have been doing a very extensive research with proteomic analysis. An important thing is that if you remove all the cells from the tissue, you see that there are many more extracellular matrix protein evident, which are usually masked by the presence of cells. And so looking at this comparison, we thought that probably scaffold could be a very good source of biomarkers research, for biomarker research. For example, this is was presented at last EASL, and there will be a big presentation about development of this at the next EASL meeting in Amsterdam. Basically, if you compare a healthy human liver scaffold with NAFLD and PSC, there are some ECM proteins which are in common, but there are very defined signatures for PSC, for NAFLD, and obviously from healthy. So we start working now on specific biomarker for PSC, which seems to be quite promising. So once again, the proposal is to use all human models with very limited animal models to try to translate better into human. So this is just to thank the people who work with me, Giuseppe, my PhD student, now he's a postdoc, and Professor Malago, the surgeon. These 3 guys, they work on intestinal scaffolds, on IBD, for IBD fibrosis. And this is Walid Al-Qad and Andrea Telese, which have been very important for the team. And this is a scaffold of almost 2 kilos discarded human normal liver, and these are the other co-workers for this work. Thank you very much.
Thanks, Máximo. This state-of-the-art lecture. There are no questions. Thanks for this fascinating and exciting data.